Hello, my name is Bill King, and I'm the Director of Research and Development and Clinical Research for Owl Manor. I have a 20-year experience working on regenerative medicine and regenerative medicine devices. I performed my PhD in biomedical engineering from the University of Wisconsin-Madison. I did postdoc research at the University of Michigan before transitioning to human health at Biomet and in Zimmer Biomet, and now at Owl Manor, working on animal health. Owl Manor is a medical device company developing and selling products designed to address injuries common to companion animals. We have a medical device portfolio that's designed to harness the patient's natural healing mechanisms found in their own blood and bone marrow, including the Pro-Stride APS device, the Restogen PRP device, and the Centrate bone marrow concentrating device. Our products are sold globally to veterinarians and veterinary practices. We were founded in February 2015, and our operations are based in Warsaw, Indiana, the orthopedic capital of the world. We have a strong 20-plus year development of history commercializing and developing these products, and I'm pleased to share that with you today. The focus of today's talk is the Pro-Stride APS device. Pro-Stride is a proprietary dual device system whose output produces a concentrated solution of cells, platelets, growth factors, and anti-inflammatory proteins. The whole process takes less than 20 minutes and can be done ambulatory stall side at the point of care. We love seeing pictures of our centrifuge on your trucks and how they're incorporated into trucks and where in the world that you've been with them, the success you've had with them too. It's been exciting really to see the broad adoption. A little more technically about what is the product. In the first device, we load in 60 milliliters of anticoagulated blood. It's placed in a centrifuge for 15 minutes. And during that process, the dual buoy system separates the blood components. The bottom buoy is tuned to the density of red blood cells. And so it floats up to the hematocrit level of your patient, capturing a high percentage of platelets and white blood cells and a small volume of plasma. After that 15-minute centrifuge spin, you can extract 6 milliliters of a highly concentrated cell and platelet solution, which is transferred into the APS concentrator device, which contains polyacrylamide beads, which desiccate the solution, drawing out water and concentrating the components further. Then a 2-minute spin is performed, in which the APS is drawn into the bottom container, allowing for 2.5 milliliters of a concentrated anti-inflammatory and growth factor solution to be drawn out. A question we were all asked to address is, are the contents of the product quantified and tested? And if so, how often and how? Well, the Pro-Stride APS device it's a dual device system, like I said, and it's manufactured under very strict quality control in an ISO 1345 certified facility in Huntington, Indiana. So all of the medical device manufacturing requirements for human health are also employed here. The scientific rationale for the product comes from the last 20 years of research into osteoarthritis. Historically, osteoarthritis was thought as purely a mechanical disease, but now we know that there's a strong biochemical component to it as well. Specifically, catabolic pro-inflammatory cytokines like interleukin-1, IL-1, or tumor necrosis factor alpha, TNF-alpha, drive inflammation and cartilage degeneration. And research has shown that you need to provide more than just anti-inflammatory cytokines or anabolic growth factors to bring this into balance. Human clinical trials of recombinant IL-1RA or IRAP have failed to address human osteoarthritis, as well as other recombinant growth factors in humans as well. Rather, we need both. And that's because osteoarthritis is a multifactorial disease and really a disease of the joint. When osteoarthritis progression happens in the way that the schematic shown here on the left, well, there'll be some trauma or genetic predisposition that causes the secretion of interleukin-1 or TNF-alpha, those inflammatory bad guys, from cartilage cells or synovial cells in the joint. those inflammatory bad guys bind onto receptors to the cells and cause the secretion of matrix metalloproteases, or MMPs. You can think of these as molecular scissors that chew up the cartilage tissue, and the fragments released bind to cells, and the cells sense them as tissue damage and secrete more inflammatory bad guys to combat this tissue damage, causing this runaway train of inflammation and tissue destruction, causing more inflammation. And so when we inject the APS solution containing this concentrated solution of anti-inflammatory proteins and growth factors, we can address that. And it acts on both the cartilage and the synovial lining because, again, osteoarthritis is a disease of the total joint, stopping that runaway train. So the goal of Pro-Stride is to provide both anti-inflammatory cytokines and anabolic growth factors to restore balance to the joint. And it's the composition of APS that makes it unique in the area of point-of-care devices. APS contains concentrated white blood cells, platelets, and plasma proteins. And I spent many hours in the laboratory pulling apart these different components and measuring what each one of them contributes. White blood cells contribute IL-1RA, or IRAP, which blocks interleukin-1. They also contribute soluble tumor necrosis factor receptors 1 and 2, or STNFR1 and STNFR2, which block TNF-alpha signaling. There's some research that reports that white blood cells are bad for these applications, but the last 10 years of research has shown that not to be the case. In fact, in human clinical trials of APS, patients who received higher concentrations of white blood cells in their APS were more likely to be OMERACT-ORSI high pain responders, meaning that if they received more white blood cells, they had greater pain relief. I'll state that again. If they had more white blood cells, they had more pain relief. And the research explains why that's so. If there's greater concentrations of white blood cells, there's more IL-1RA. And in fact, in my research, I've seen repeatedly that increasing concentrations of white blood cells actually result in higher IL-1RA to IL-1 ratios, showing that they don't contribute inflammatory bad guys in this context when processed through the APS device, that we have more anti-inflammatory good guys and less inflammatory bad guys. Platelets contribute anabolic growth factors that are important for cartilage health, like TGF-beta and PDGF and FGF2. Concentrated plasma contains both anti-inflammatory cytokines, like soluble interleukin receptor 1, another way to block interleukin-1 signaling, as well as those STNFR proteins we talked about, and A2M, because it's a concentrated plasma product. Also, there's anabolic growth factors like IGF-1 and HGF, which are important for cartilage health. So together, the concentrated white blood cells, platelets, and plasma proteins are what enables the output of the Pro-Stride APS device to be unique. And it's not me just saying that in a cartoon. We have extensive research to back that up, starting with cells in a dish, scaling up to explants, scaling up to research models of osteoarthritis, and finally into models that are relevant to your patients like equine and canines with naturally occurring osteoarthritis. And I'd like to touch on some of the highlights of this published research today. And we've done these in partnerships with great research partners all over in academic and private labs. And I just want to say that they've been contributed greatly to the success of the program. When evaluating new osteoarthritis modalities, a place that I like to start is this chondrocyte assay, where cartilage cells are grown in a dish and challenged with inflammatory cytokines like TNF-alpha and IL-1. And then we measure the amount of MMP13 or collagenase they excrete when challenged with these inflammatory proteins. So the more MMP13 on the y-axis here, the more tissue destruction we would expect. And in the study here, we showed that if we had TNF-alpha, we saw some MMP13 production. If we added interleukin-1, we saw even more MMP13 production. And we added TNF-alpha and interleukin-1 together, we saw the synergistic response where even more MMP13, more tissue destruction would be caused from those. However, when APS was added, we were able to successfully block TNF-alpha alone, interleukin-1 alone, and their combination together, showing that Pro-Stride APS blocks multiple inflammatory pathways. Scaling up from cells in a dish now into explants, we took bovine cartilage explants and challenged them with interleukin-1, one of the inflammatory bad guys. And you can see in the untreated cartilage explant, you can see individual cells in a dish by themselves in their lacunae. When we added recombinant IL-1RA, the drug form, in a very high concentration, we saw the same response, that there was still these individual cells in a dish. However, when we added APS, we saw that there was actual cell division. So we saw a regenerative response in response to the APS. We also saw that there was decreased release of glycosaminoglycans and collagens from the cartilage explants in response to APS in these inflammatory conditions, showing that APS allowed for better cartilage health in these inflammatory environments than the recombinant IL-1RA alone, showing again here that it's the soup of those anti-inflammatory factors and growth factors having an effect. Scaling up again now, we started with cells in a dish, then to explants, and now we're looking at the total joint. And a common way to do this when looking at new osteoarthritis modalities is the meniscal tear model. So instead of tearing out the whole meniscus, a tear is made, which creates this graded osteoarthritis over the course of a month in rats. And a place that's commonly done is at Boulder Biopass with Alison Bindiel, who's a leading scientist in this area. And when I talked to her about these experiments, that I wanted to have it be a single injection, she laughed at me, kind of like, that's cute, because they do drugs that are fed every day, or products that require a series of injections. But in this case, I wanted to do a single injection because we thought that would be important for the development of the APS device. So in this experiment, some rats were left without surgery as a control. Some were injected with saline and some were injected with APS 1 week after the meniscal tear. Then they were allowed to walk around for 3 more weeks to continue to develop osteoarthritis, and they were sacrificed, and then there was macroscopic and microscopic evaluation 28 days later. And average micrographs of these knees are shown here. And you can see that in the non-surgical control, in A, you see a smooth articular surface. In rats that were injected with PBS, we see that the cartilage there's great cartilage destroyed. We don't see that smooth articular surface anymore. And in C, we see that there's still cartilage present, but the intensity of the staining is less. What we don't know from the output is that was there a regenerative response, or did we change the course of the disease progression and slowed it down? And I think it's likely that latter case where we changed the course of the disease progression, but further research would be necessary to show that definitively. Also, this paper showed that we had improvements in synovial tissue and subchondral bone, allowing for an improved total joint score with the APS-treated rats compared to the saline-treated rats. Again, showing that osteoarthritis is a disease of the total joint, and APS is having an effect on the total joint. Scaling up again, here we see we did a equine clinical trial in partnership with Alicia Bertone at Ohio State, in which 40 horses with naturally occurring osteoarthritis were brought in and maintained at Ohio State for 2 weeks. 20 received injections with Pro-Stride APS, 20 received injections with saline, single injections. And then there was lameness scoring by AAP lameness grade, as well as force plate analysis, and they were exercised on a treadmill every day for the duration of the study while they were at Ohio State. And we can see that horses that received Pro-Stride APS had significantly improved lameness 7 and 14 days after injection compared to saline control. The results were mirrored well with the force plate analysis in the study. After these 2 weeks, owners of the horses who received saline control were offered Pro-Stride APS crossover treatment. All of the owners elected to do so, and 40 horses were sent home after receiving a single injection of Pro-Stride APS. Owner surveys were sent out 3 months and 12 months after injections. And here we can see that there was significantly improved degree of lameness, comfort at rest and stall, and comfort at turnout after that single injection, 3 months and 12 months after the APS. And this now has been mirrored in a broader clinical experience. So to answer the commonly asked question, how many patients have been treated with this product in animal health? There's been tens of thousands of devices used in the product family, including Pro-Stride APS, Restigen PRP, and Centrate BMA. In human health, there's been hundreds of thousands of these devices used at Biomet and now Zimmer Biomet. So there's a long history of experience to draw on from these products. One of the questions we were asked to answer is, have adverse events been tracked? If so, what are results and how are they tracked? We track product complaints in line with the ISO 13485 medical device requirements, and our product complaints are far below 0.1% of all products sold. The data that we have following for efficacy is the Bertone study reported to us on significantly improved lameness in 2 weeks, and that durability is, as reported by owners, 1 year post-injection. And this has been mirrored by our clinical experience in talking with the hundreds of people who now have Pro-Stride in their practices. A nice discussion of this happened at AAP in 2019 in the Regenerative Medicine Forum, in which Dr. Tim Ober stated that in some of the horses he had originally been addressing with ACS or IRAP products every 3 months, when he transitioned to using Pro-Stride APS, the duration between injections got longer. And with each subsequent injection of Pro-Stride APS, the injection time got longer, then even more. And earlier this year, I was at a Equine meeting in Jumping Amsterdam, or in parallel with that. And similar results were also reported at that meeting as well. And so this durable clinical response has been seen not just in the Ohio State study, Dr. Bertone, but also in practice with the hundreds and hundreds of people who now use Pro-Stride routinely. A question that we were asked is to describe the sort of treatment and the aftercare. Is it a series of treatments? The published research in horses and now in the 2 randomized controlled studies in dogs show durable clinical outcomes from Pro-Stride APS after a single injection. And reports to us on treatment and aftercare vary differently based on the veterinarian, the discipline, the condition of the horse, etc. So it's really hard to say an individual thing. That being said, now that there have been hundreds of people who use the Pro-Stride system, there's lots of great expertise to draw on from your fellow equine practitioners. If you have questions, please don't hesitate to reach out to Owl Manor, and we can put you in touch with someone who has the appropriate experience, and you can have that conversation together. So what makes the Pro-Stride product unique is that Pro-Stride has a unique combination of concentrated platelets, white blood cells, and plasma proteins. And together, this allows for it to have a uniquely anti-inflammatory profile while also containing growth factors. It takes less than 20 minutes to process, so it can be done anywhere, anytime, ambulatory off the back of your truck, and it's all contained and sterile. There's extensive research backing up the data presented today, including 50+ peer-reviewed publications and abstracts from meetings with clinical support and coupled with 20+ years of manufacturing expertise. And to unpack that a little bit, we wanted to make sure that the output of the device and the development of the device was part of the normal peer-reviewed scientific process. So there was a concerted effort to make that point. So the research is published in mainline orthopedic journals like the Journal of Orthopedic Research. There's just a huge body of evidence to fall back on there. And when I had transitioned from Zimmer Biomet to Owl Manor, I was talking with an equine practitioner who I knew had done work with a competitive company in the past, but I found out she was using Pro-Stride, and I said, "Oh, what brought you to use Pro-Stride?" And she said that there's no better characterized device output than APS. And that's what gave her the confidence to switch from what she was previously using to using APS. And that really gave credence to the hard work that we have been putting in both preclinical and clinical research. And the 20 plus years of human experience and veterinary experience together allow us to have a deep and institutional knowledge of all the different ways that these products have been used in human and animal health. And it allows us to have a body of knowledge that no other group has. And finally, APS has been clinically proven in randomized and controlled trials and in tens of thousands of equine cases. And that's important because I find in a lot of these panel discussions that people will focus on an individual concentration of an individual factor. And as a lab guy, I get that. I care about how things are measured in the lab, et cetera. But in the end, what matters is durable clinical results and the investment and the evidence that exists. And Pro-Stride has that. And that there's now tens of thousands of cases. So if you have questions that come up, there are people that you know, that you trust, who have likely had the kind of experience that we can fall back together on and keep building those success stories together. And so it's really this combination of very detailed technical development, published research, and clinical expertise that makes Pro-Stride unique. With that, thank you for your time. If you have any questions, don't hesitate to reach out to contact us. You can reach us through various social media channels, through our website, owenveterinary.com. And for more specific information on Pro-Stride itself, you can visit prostride.com. Thank you for your time, and I look forward to discussing Pro-Stride with you further.